Welcome back to Chapter 21, Part 2. In this part, we are going to be doing some math. It might help if you have a calculator ready so that you can play along with some of this math. But before we dive into the actual math itself, we do have a lot of terms to kind of understand. So I emphasized it very much in the last lecture here in Chapter 21, Part 1. The genetic variation is absolutely required for a population to evolve. But it does not guarantee that evolution will occur. There has to be a cause for evolution. There has to be a reason for that allele frequency to change, for that population to evolve. And and you can see some of the math that we're going to be getting into. But first, let's do some terms. A population is a group of individuals that live in the same area. That's actually really important. They live in the same area and they interbreed. Actually, this interbreed concept is part of the definition of species. So basically, they also have to be able to produce fertile offspring. So basically, what we've got is members of a species living in the same area. Again, species is defined as whether they can interbreed and produce fertile offspring or not. So populations don't have to be geographically isolated, but if, like this example of these different herds here found in overlapping ranges, but it turns out that the members of the porcupine herd, and no, they're not porcupines here, that's just the general area where they're found, they only really interbreed with each other. So even though the range overlaps right here, the porcupine herd does not really mate with the 40-mile herd, and so they are considered to be distinct populations, again, because of this overlapping non-interbreeding, and so forth. So, and just so you know, these are, which I didn't say before, these are reindeer, and they are found at the border of Alaska and Canada. So another term that's really important to understand when we're talking about the math of evolution is the gene pool. And the gene pool consists of all the alleles for all the loci in a population. Remember, loci, that's plural for locus, and a locus is just a location on a chromosome. So this is a good way to think about it. Separating the alleles from the individuals that hold those alleles. That's a really important thing when we're talking about evolution. Just this intellectual thing. Well, this one's big B, little b, but all I'm thinking about is the gene pool. Again, another way to define gene pool, which I like, is it's the combined genetic information of all the members of a population. So we're just combining them in this pool here, right? Another term that's important for understanding evolution is that sometimes an allele for a particular locus is fixed. And what this means is that all the individuals in the population are homozygous for the same allele. Now, why is that important? Because remember, the evolution requires variation. And if the allele is fixed, then evolution can't happen. So yeah, another important concept in everything that we're going through, which is we're going to start diving into the math really soon, because this next term that I have for you is frequency of an allele. Another way to say that is allele frequency. So you'll see it written both ways. So how do you calculate allele frequency? And the important thing here is to remember that for each individual in a population, they have two alleles. So if you want to know what the total number of alleles are in a population, you're going to take each number of individuals, right? And you're going to multiply that times 2, because there are 2 alleles for each individual in the population. So the total number of alleles is equal to the total number of individuals times 2. The total number of dominant alleles can be calculated by looking at the number of individuals that are homozygous dominant and the number that are heterozygous. And the way that we do this is, if I'm only interested in the dominant allele, then I will count up how many individuals are homozygous dominant and multiply that times 2. But I'll count up the number of individuals that are heterozygous, and I'll only multiply that times 1. Because in a heterozygous individual, only one of the 2 alleles is the dominant allele. And I can do the same thing for the homozygous if I want to calculate the number of recessive alleles. I'll take the number of recessive individuals and multiply that times 2. And then I will take the number of heterozygous individuals and multiply that times 1, because again, there's only one recessive allele in those heterozygous individuals. When we're just learning to study evolution, we take the simplest version of this. And so we're only going to assume that there's one dominant allele and one recessive allele. Clearly, we've learned that that's not always the case, but that's the way we're going to treat it for now. So if there are 2 alleles at any particular locus or gene, then we use the letters P and Q to represent the frequency of each of those. So P represents the frequency of the dominant allele and Q represents the frequency of the recessive allele. And yes, we do always use P and Q. Those are like our constant letters. Since we're talking about frequency, and frequency is a subset divided by a total, right? So if we're talking about frequency, then if you add up the 2 subsets, you're going to get a total of 1, right? 1 out of the total 1. So P plus Q always equals 1. That's also a good double check if you're trying to figure out if your math is working out, because if P plus Q doesn't equal 1, then you might have done something wrong. Let's take a look at an example of some of the things that we've talked about here and really dive into the math. So, and we're picking a particular trait that's easy to visually see what the genotype is. So here we're looking at something that has incomplete dominance. We like incomplete dominance for our initial example because we absolutely know that the heterozygote is got both alleles and they're showing this intermediate phenotype. Okay, so again, you might want to get your calculators out and go through some of this math with me. Let's say we have 320 red flowers. They're going to be homozygous for the red allele. And 160 pink flowers, right? They're going to be heterozygous. And 20 white flowers. There they are, homozygous for white. Notice I didn't say dominant and recessive, but let's just for argument's sake say red is dominant. We're going to give that P. Again, just because of conventions, right? Like we only have P and Q, and P is defined as that frequency of the dominant allele. When we're dealing with incomplete dominance, we don't have a dominant allele, so we just arbitrarily assign it to P or Q. All right, so the first thing that we have to do is to calculate the number of copies of each allele. And I stepped you through that on the last slide, or 2 slides ago, I guess, where we take the number of individuals who are homozygous for red, and we multiply that times 2 because each red individual has 2 red alleles. So 320 times 2. And then we take the number of individuals that are heterozygote, and that's 160. Now remember that only one of those is the red allele. So you could say just plain 160, or you could say 160 times 1. Either way works, especially if you're really just confused about when you multiply times 2 and when you don't multiply times 2. So 320 times 2 plus 160 gives us 800. And we can do the same thing for the white allele. There's 20 white flowers, so 20 times 2, right? Because there's 2 white alleles here. And then 160 times 1, because there's only 1 white allele in there, gives us 20 times 2 plus 160. That gives us 200 alleles. And that's the beginning of the math. That's just how we figure out the number of each alleles if we are given the number of genotypes of each type. So how do we figure out P and Q from this? Well, the first thing that we can do is we can calculate the frequency of P. And so P here, again, we're arbitrarily assigning P to the frequency of the R allele. So we do the subset, which was 800. Remember, over here, 800 alleles, divided by the total. Now, the total is 800 plus 200 because we have to do the total number of alleles, 800 plus 200. So 800 divided by 1,000 is equal to 0.8, which we can say that's equal to 80%, but we remember that we always want to keep in mind that P plus Q equals 1. So we're really going to focus on this decimal right here. P equals 0.8. For Q, the really easy way to calculate Q, as long as we're really satisfied that we've calculated P right, is just to remember that P plus Q equals 1. And if P plus Q equals 1, then 0.8 plus Q is equal to 1. And if 0.8 plus Q equals 1, then Q must be 0.2, right? That's the only way to get to that number. So P plus Q equals 1. And again, if you do it with percentages, the math doesn't work out quite as nicely because you still have to convert it. P plus Q equals 1. This is basically what I'm showing you is something called the Hardy-Weinberg equation. And this is named after 2 dudes back in I think it was the 1920s, like 100 years ago, who figured out the math of this. They did it individually, and then they published it about the same time. So we call it the Hardy-Weinberg equation. This equation describes the genetic makeup of a population that is not evolving. So important. We want to have like a null hypothesis, right? So the Hardy-Weinberg equation is basically a null hypothesis. And so what they would say is that if your population, your P and your Q stays the same from one generation to the next, then you got a population that's not evolving. Remember that the definition of evolution is change in allele frequencies, and P and Q are allele frequencies. So if the data differs from the expected values, right? Remember, we were expecting Q to equal 0.8 and P to equal 0.8 and Q to equal 0.2. If it differs from that in the next generation, then we might be having some evolution going on here. And we're going to talk a little bit more. We're going to go into some depth here on the Hardy-Weinberg equilibrium. Again, I kind of talked in the last slide about the Hardy-Weinberg equation, and we're going to go into that into a lot more detail again. But Hardy-Weinberg equilibrium says that a population, which is mating at random, and none of the mechanisms of evolution, which I mentioned 3 of them before in this lecture. Actually, in part 1, I think I mentioned them in part 1. If none of the mechanisms for evolution are acting, then we say that the population is in Hardy-Weinberg equilibrium. That just means no change. So if you've got butterflies here, and they go through generation after generation, and there's no change, then this population is in Hardy-Weinberg equilibrium. Another way to say this, and it's actually better than this example, because in this example, we're talking about genotypes, although we can absolutely calculate, we've just learned how to calculate allele frequencies from given genotypes. But another thing that's really important, and this example isn't so great, is because it shows that the actual numbers stay the same from generation to generation. That's not as important as P and Q staying the same. Remember that P and Q are allele frequencies, and those are proportions. Another way to say this, because I think I might be confusing a little bit, is let's say you take a quiz and you get an 8 out of 10. Or you take a test and you get 80 out of 100. You still got an 80%. Doesn't matter if you had 10 questions or if you had 100 questions, you still got an 80%. So your percentage stays the same, right? So your grade isn't changing. So this is just a very, very simplified way of thinking about them with always being out of 100%, for example, on a test. So if allele frequencies remain constant from generation to generation, we got our Hardy-Weinberg equilibrium. And so let's take a look at a little bit more closely this concept of Hardy-Weinberg equilibrium. All right, so we're going to start out with the ideas that we've talked about before, right? We're talking about alleles that are consistent in frequency in a gene pool. And let's say that we have a population of 500 wildflowers and there are 1,000 alleles, just like we were talking about before. And we're just going to step through this one by one. So the allele frequency, as we calculated before, was 0.8 for the red allele and 0.2 for the white allele. And then you throw those alleles into a gene pool. Again, you're not literally throwing them into a gene pool, but we are going to say that the population is mating randomly, right? So if the population is mating randomly, then it's kind of like throwing these alleles into a gene pool. What you would expect is 80% of those alleles would be the red alleles and 20% of them would be the white alleles. And then, of course, if you do have random mating, then you're going to randomly pull out. You got an 80% chance of pulling out that red allele and that 20% chance of pulling out that white allele for both the male and for the female. So in the end, what you have is a population that looks just like the original population of alleles. We can actually use the concept of Punnett squares to figure out what the genotype frequency is going to be. We're using this kind of prediction. We can predict the frequency of genotypes by using the rules of multiplication like we learned about for Punnett squares. So if your red allele is at 0.8, then your chances of having an offspring that's homozygous for red is going to be 0.8 times 0.8, right? Or 0.64. And we give that the name p squared. Hopefully that makes sense to you. P squared is p times p squared. And p squared also represents the frequency of the homozygous, in this case for red, or we could say homozygous dominant if we were actually using dominant recessive in our example. Now, if we're looking at the heterozygous, there's actually two different ways that you can get the heterozygous. You can have the sperm having the white allele and the egg having the red allele, or it could be the other way around. You could have the egg having the white allele and the sperm having the red allele. And that's like multiplying P times Q. If I want to know the chances of having a heterozygous this way, I multiply P times Q. So 0.8 times 0.2 gives us 0.16. Oh, and also, we have to do it this way as well. So 0.8 times 0.2 gives us 0.16. And then we have to add those together. So we get 0.16 plus 0.16 is 0.32. Another way to calculate this is to simply acknowledge that there are two different ways, and to do P times Q. And so we can just say 2 times P times Q. And that gives us the frequency of the heterozygous pink flower. If we want to calculate the frequency of getting the white flower, you would do the frequency of having a white allele in a sperm and a frequency of having the white allele in the egg. It's 0.2 each time. So 0.2 times 0.2 is 0.04. And that's like saying Q times Q, right? So Q squared. So what we have defined today in this moment on this slide are some really important concepts of P squared, 2PQ, and Q squared. These are the frequencies of the various genotypes. And we can define these terms in this way. Just remember that P and Q represent the relative frequencies of the 2 possible alleles in the population. And by doing all that math that we did on the previous slide, we get P squared plus 2PQ plus Q squared. And just so you know, if you add all those up together, you do get 1, just like with P and Q equaled 1. P squared is defined as, just like we defined P earlier, we're going to define P squared as the expected frequency of the homozygous genotype. I know here it says of the CRCR, but we're going to say homozygous dominant. And that's because we define P as the frequency of the dominant allele. So P squared is defined as the frequency of the homozygous dominant. 2PQ is defined as the expected frequency of the heterozygous. And Q squared is defined as the expected frequency of the homozygous recessive. Again, I'm totally acknowledging that this is not recessive, but it was useful in our example to be able to see the heterozygotes. So that's why we did the incomplete dominance. Again, just one more time, P squared is defined as the frequency of the homozygous dominant. 2PQ is defined as the expected frequency of the heterozygous. And Q squared is defined as the expected frequency of the homozygous recessive. We are going to be visiting this math again in part 3, but we're also going to be applying this to the conditions of the Hardy-Weinberg equation or Hardy-Weinberg equilibrium, and we're going to be applying it to the idea of the causes of evolution, like what causes evolution to occur. So I'll see you again in part 3 of chapter 21.